So it's great to be here. Thank you very much for the invitation, for the introduction, for hosting me. I think you have an amazing environment here, and I really enjoyed so far very much, my stay. So I was asked to give a few minutes about some anecdote or some perspective, I guess, on how I think about what's something unique about the way, more personal about science. And I think when maybe something that we don't really talk about in science, when we think about the scientist, we think of someone who is very kind of hyper critical, very, how you could say, everything is statistical, very precise, very rigor, right? And of course, this is how we want science to be. But I think maybe one of the things that pushes science forward more than anything else is something that I think call optimism. I think it's really about optimism, the ability to even look in the results and I think mainly in fields that, like the one that I work at, that I don't have an idea where I'm going. Things are really unclear. And most of the time, the first results are, if I'm really lucky, they are in the neighborhood of the right phenomena. And so no way it's going to be statistically significant in the beginning. And I think it's very hard often for students but this is not how we train. But I think, and then the art is actually in identifying maybe sometimes it will manifest as the standard deviation has changed. And what you're looking for is actually, it's not that I see a statistic, but I just see something different in the standard deviation. Or you suddenly start to get outliers that are, it's weird. There is something happened biologically in them, but you don't have an idea. It's definitely not statistical. And it's almost like, I think, gold mining, that you're looking for these glitters of gold. And most of the time, they are illusions. And it takes really, I think there's some level of resilience to students to be able to handle, and myself as well, to handle all the disappointments in following these glitters of gold. But I think this is also part of the exciting thing. And I think we shouldn't, sometimes we feel that this is kind of a disadvantage, that you are, you're hopelessly romantic, I would say. But no, this is what makes science, I think, a beautiful thing. And well, yes, when the field is really mature, you can go like in a gold mine, you can go for the big chunks of gold and you know exactly where you're looking for and then do the big discoveries. But I really hope that I wish you a lot of glitters that you follow and turned out to be a real thing and not just illusions. So I guess this is my perspective on the scientific discovery process. Moving to science, to my science. Okay. Thank you. So I will talk about actually a glitter of gold, if I'm thinking about it. 1800 London, huge sensation. They created a machine that you plug to the patient and it pulls out the disease out of the patient. Every kind of disease, obviously it's ridiculous, but it's working. Many people are actually getting cured by this machine. So there was a very famous physician at the time, John Hayward, and he was a bit skeptical about this new phenomenal machine. And he built what we consider now the first placebo-controlled trial. He built a machine that looks exactly the same, makes a lot of noise, but it didn't have this special engine that pulls the disease out of the patient. And of course, as you can imagine, it proved that the machine doesn't work. But the way he summarized it, I think it's amazing. An important lesson in physics is here to be learned. The wonderful and powerful influence of the passions of the mind upon the state and disorder of the body. It's there all the time. And we just need, I think, to really start to work on that. And we see it in this mind-body connection. We see it all the time. When we have a deadline and we don't get sick before the deadline, or when we are sick, we get a throat infection and suddenly the world looks gloomy. Why? We don't communicate. So the connection is always there. It's just a question of how. So in epidemiology, we have huge connections between stress and any kind of disease. In medicine, we have all this field of psychosomatic disorders, things that we basically have no idea what's going on with the patient. The clinicians have the feeling that it's somehow related to their emotional state, not much to do with the patient itself. And in pharmacology, we have this placebo effect, right? The small, tiny thing that we give a sugar pill and people are getting better. And it's a huge problem, actually, for pharmaceutical companies because if it kills a lot of pharmaceutical experiments. But what I want to say that it's really not just a problem. It's actually a huge opportunity. Because if we start to think about it, if a person gets a sugar pill, there's some process that happens in their mind. There is like positive expectation, hope, something that that somehow then changes the state of the body. If we just understand that, how it happens, we can harness it and now start working with that. And I think the big transition that we can do is by taking this mind and really big conceptual psychological existence into brain activity and see what is active in these cases. And one of the things that we know is that reward system is activated during positive expectations. So we are going to ask how reward system activation affects the immune system. So the reward system, and this crowd doesn't need the introduction, is this system that is involved for positive expectations, hope, motivated behaviors. It's a key component of it is this WTI dopaminergic neurons that project throughout the brain. And the question that we wanted to ask is how reward system activation, specifically these WTI dopaminergic neurons, affect the immune system. And it's a very reduced question that I'm going to ask. We're going to ask it in mice. So the first one, and I'm going to show you briefly some of this data, really briefly, and then go to some of the newer things. So what we did in this work by Tamar Ben Sha'anan, actually now she is a PI at the Weizmann, together with Shai Shen-Or's lab. What we did, we activated the reward system of mice. So we used dreads, which allow us to locally activate these neurons, basically by targeting specifically, locally injecting the dread to the VTA, and genetic transgenic mice that allow us to target it to the dopaminergic neurons. We have a control, of course, for the surgery, for the viral infection, for the CNO injection that is required to activate the dreads. And then we are going to have two groups of mice. One group in which we are going to have a control, and one in which we are going to activate the VTA. So just to show you some vignettes of that. If we take these mice, activate the reward system, and then extract from their blood immune cells, specifically monocytes. And what monocytes can do, one of the things they can do is to kill bacteria or to phagocyte it. So we are just going to incubate these immune cells that we extracted from the cells with E. coli, and then plate them on these petri dishes to see how many bacteria survived. So what you can see here is this is a control mouse, and this is after VTA activation. So you can see that there is much less bacteria surviving after this interaction, meaning that it somehow boosted the immune response. Can we also see it in vivo? For what we did, again, we activated the reward system on control. Now we injected E. coli fluorescent bacteria. So if macrophages are going to phagocyte it, they are going to become green. So we are looking for how many GFP positive phagocytes are there. And you can see it here. This is a fax plot. You can see that in the VT-activated mice, and you can see it in the summary, there is much more bacteria, macrophages that actually phagocyte the bacteria. So we can see there is some kind of activation that is induced by this reward system activation. And the question that one should ask is why? Why should the brains, why should the reward system even have anything to do with immune system activation? So one of the ideas that we came up with is that basically what are natural rewards? When is the reward system activated? What are natural rewards? Feeding and mating. And both actually expose you to pathogens. It's interaction with someone else or with food. So if you prime the response immune system before, then you might have an evolutionary advantage. And indeed, And so a few years ago, it was shown directly by another group. And what they showed, so they had mice. So apparently, I didn't know about it. But apparently, mice that are anticipating mating have elevation in different cytokines, anticipating mating. If they block WTI activity, they lose this natural increase in cytokines, suggesting that indeed the reward system is actually involved also in natural processes of immune regulation. Okay, but of course, the immune system is not only for fighting bacteria, it's also involved in many different processes. And Dr. Faid Hakim, who is the head of Nazareth Medical Center, came to me and said, well, actually, it might be even relevant for cancer. So this project was then done by again, Tamar and Maya Schiller. She is an MD, PhD, now mostly MD, and with Mickey Rath. So what we did is the following. Again, now we are going to inject mice with tumors. So we have LLC model or B16 melanoma, and I'm going to show you just the Lewis lung carcinoma, the lung one. So we're injecting mice with the tumors. We have, again, these two groups of mice in which in one we can activate the reward system, in other not. And for the 14 days, as the tumor grows, every day we are going to activate the reward system. And then we're going to look on the tumor size. And this is how it looks like. So you can see here in the top panel, you see tumors from control mice. And in the bottom one is from VT-activated mice. And we did not have any intervention in terms of beside reward system activation. So how did it happen? Because I'm doing the manipulation in the brain, in the VTI, but the tumor is in the periphery. So how does the signal go by? It can be dopamine, in principle, because I'm manipulating dopaminergic neurons, but dopamine doesn't cross the blood-brain barrier. So what we found was that the signal was actually mediated via the sympathetic nervous system. So if I'm talking about the communication from the brain to the periphery, we'll have sympathetic, parasympathetic. And these maps that I take from all the classical textbooks actually are really incorrect. Because one of the things that we now know is that the sympathetic nerves go to all the immune organs. They innervate all the lymphatic system, the lymph nodes, and they actually play a very important role in this process. So how, in this case, how was the signal mediated? We found a reduction in sympathetic activity, specifically in the bone marrow. Why bone marrow? Bone marrow is a really cool site, actually, for the brain to communicate. And I can tell you that a lot of the data that we have is that the brain communicating, the more we look into it, a lot of it goes through the bone marrow. Why bone marrow? Bone marrow is where we have our hematopoietic stem cells. So basically, this is where we keep our immune stem cells. They will regenerate the entire immune system. So the brain speaks to the source. The brain actually educates the nature of the immune cells that are going to be formed in the immune system. And indeed, one of the things that we found, that a subset of immune cells that is really critical for the cancer, because what the cancer does increases the level of these immune cells that are immune suppressive. And actually, when they're in the bone marrow, they have high levels of beta-adrenergic receptors. So they're even tuned in the bone marrow to listen to these guiding signals from the brain. And in fact, when they get, so the sympathetic activity actually regulates their activity. Now it releases the brakes from the immune system, and we have stronger anti-tumor immune response and reduction in the tumor size. So at this point, another clinician came in. Lior Gepstein, he is head of cardiology. And he said, well, you know, I have a lot of patients that come with a heart attack. From my clinical perspective, they all have exactly the same heart attack. I have no idea how to, but one is going to recover and the other is going to completely decline. And intuitively, I think that it's somehow related to their personality or attitude or something of that sort. So Chetva Chaikin in the lab, she was a joint PhD student, started to look into it. And apparently, the American Heart Association actually looked at that in meta-analysis. And what they found is actually that high levels of optimism are correlated with reduced cardiovascular disease risk. So back to the idea, optimism is not only good for science, it's also good to your heart. So another thing, another trait that they found to be correlated, which is really cool, is sense of purpose. I think in science we are lucky to have a lot of that. So what Hedva did, she actually took and did this, again, the same paradigm you already know, WTI activation controls. She did a heart attack. So she actually had an AMI. You have a ligation of the LAD. Because there is a lot of variability in the surgeries, they're super complicated surgeries, so we test every mouse in MRI to make sure that they have the same range of damage to the heart. And then for 14 days, as the mice recover, she's going to activate the reward system once a day, and we're going to look on the heart recovery. So what you're seeing here, it's a slice of the heart at a certain area. This is a normal heart. From the same area, in hearts that went through a heart attack, you can see these are 4 different mice. You can see that a visible damage, right? After VTA activation, the same mice that went through a heart attack, this is how they look like. So you see, it looks very different. It looks more like the normal mice. And we also tested that in MRI. So basically, it's left ventricle ejection fraction, which would mean how well the heart pumps blood. And what you see in this model is that usually mice that had the heart attack, after time, they will have a reduction in heart activity. But in the VT-activated mice, most of them did not get worse over time, and some even got better. We didn't have enough mice to do the comparison to see what happened to those who actually got better, so it was too small of a sample size. But I think it's really interesting to see what these outliers are. So how did it happen in this case? Again, sympathetic activity. Again, a reduction in sympathetic activity. So of course we look in the bone marrow. No, it's not in the bone marrow. Now the effect was in the liver. So I said, well, what's going on in the liver? The liver, and I became obsessed with the liver. I'm not going to show this data. We have like now a whole study on the liver. The liver is a really cool site because you have this organ that secretes proteins and peptides in different combinations, things that regulate blood flow, they regulate immune activity, they regulate metabolism, everything. So think that you have an organ that can synchronize, get messages from the brain and synchronize different responses. And what we see in this case is that the liver had a change. So first we saw a reduction in neuroadrenaline levels in the liver. Okay. And then we also saw that there was an increase in specific protein, CF3. CF3 is a component of the complement. We had no idea what to do with that. But apparently, one of the interesting things about the complement is that it secretes, it's a classical immune factor. But now we know that it's involved in synaptic pruning. We now know that it's involved in angiogenesis and many different processes. And it was also previously correlated to heart attacks. So we wanted to test whether it's involved in some way. So what we did, we repeated our experiments, VT activation and controls. But now we had, in one group, we kept the CF, we didn't touch the CF3. In another group, we actually blocked CF3. And you can see that now, just by eliminating CF3, we lost the beneficial effects of the VTA. So it seems that it also related, it's critical for this improvement recovery. I do want to emphasize that it's really not the only thing. So the fact that I focused on CF3, it's because it was a kind of very clear molecule. We also did a lot of in vitro. It seemed to affect the whole process. But I don't think that the brain activates every time only one component. So again, I really want to be cautious about it. In this case, one of the pathways that we saw is that it was mediated by vascularization, and we can even see it in the hearts in vivo that there was a change in the vascularization. So what did I tell you so far? basically that by VT activation I can cure everything. But it's an interesting question. So it seems like if I'm activating the VTA, I'm pushing the system, and I think it's related somehow to this motivation, the allocation of all physiological resources towards a certain process. And in every case, we saw reduction in sympathetic activity. It was mediated by the sympathetic activity. It was a reduction in sympathetic activity, but there was a big difference. In one case, it was mediated by the spleen. In the other, via the bone marrow. And in the other one, via the liver. So what does it mean? Is that I activate the reward system, right? But then I can get different outcomes. The brain can choose different pathways to initiate some kind of processes, meaning that, and it depends on the physiological state of the organism. So the question here, does the brain know what happens with the physiology in order to allocate that? I'm going to take a step back. I just want to say that one of the things that, another question that is very interesting here is how, if I'm doing the same activation of the VTA, how come that I'm getting every time different outcome? And we have a whole study, again, that I cannot, I don't want to overwhelm you. But what we see is the different projections from the VTA, so to the nucleus accumbens, to the amygdala, to the prefrontal, they all have different immune outcomes and actually different proteomic signatures, which will also make sense because they are involved in different processes. So of course, the physiology should be adapted differently to each one of these cases, and we tracking them down, and they seem to kind of converge differently in the level of the hypothalamus, these different pathways. So it's another. But if I'm back to the question, does the brain know what happens to the immune system? Tamar Koren in the lab, and she's also MD, PhD. She's now mainly PhD, MD, and doing some research as well. Together with Kobe Rosenblum's lab, wanted to ask this exact question. So we know that the brain responds to immune activity. Just to give you some examples, we can show that there's activity. It's known that peripheral activity is represented in the brain in fMRI studies, in meta-analysis. There's even something cooler. You can, based on the brain activity, you can actually predict whether anti-TNF therapy works before you see the clinical symptoms. So it's an interesting perspective on the whole thing. And anti-TNF are very common therapies. They're actually mostly used to rheumatoid arthritis, to Crohn's disease. So we kind of follow up in this direction. And we decided to to ask the question of how does the brain respond during gut inflammation, Crohn's. And the reason we went there is because, of course, the gut is highly innervated and a very active immunological site. So it begs to be tested. For a second, we know that patients with Crohn's they report that they know what triggered the disease, the relapse in this case. So it's highly psychosomatic connections there. And there is a reasonable animal model for DSS-induced colitis. So how do we work? We are giving them DSS in the drinking water. DSS causes damage to the epithelial cell wall, and the ensuing immune response is a typical one. So it's a model for DSS of colitis. So the question that we asked was the following. If we induce this gut inflammation and look in the brain, what can we see? We use trap mice. So trapping allows us to capture neurons that were active at a given time point. And the neurons that were, and we are doing that with the model of trap mice, which I'm sure most of you are familiar. But basically, we are capturing neurons that were active at this time point. Capturing meaning, in this case, that I can express a fluorophore in these mice. And what you can see is the red are the active neurons. And you can see that many brain areas are actually more active once we have gut inflammation. We decided to focus actually on the insular cortex. Why insula? Because it's a site of interoception. How do I feel is exactly that. So then you say, okay, so immune information is exactly part of how do I feel of this representation. So and actually it was also shown in the past that lesions to the insula can disrupt immune conditioning. So now we wanted to ask, the fact that there are more neurons active doesn't mean much, right? The question, is there a function relevant to that? So what we did, we created inflammation. The colon inflammation was DSS-induced colitis. We trapped the neurons in the brain. And now in the trapping, we actually express DREADS, GQ, so we could activate these neurons whenever we want. And ask now, after what will happen? So the paradigm is the following. We are creating colon inflammation. We are capturing neurons in the insula. And then we will have 3 groups of mice in which we are doing that. One is the group in which we are capturing the neurons in the peak of the inflammation. One is before, and one is after. So we have 3 groups. All of them, we are going to activate neurons. The difference is when we capture the neurons. We're going to wait until the mice fully recover, and then we're going to reactivate these neurons. And this is just the paradigm. You can see that we have mice captured in 3 different time points. And when we look on the colon, what you can see, the staining in red is a staining for immune cells. So you can see that only once we reactivate the neurons that were captured in the peak of the inflammation, we're actually getting reactivation of the immune response. And so it says that somehow the brain replaced this colon inflammation. So, but maybe it's only a gut-brain axis thing. So we now created inflammation in a completely different site in the peritoneum, different model. So it's we have a zymozyme, so it's a different immune response. And we again capture neurons before or during the inflammation. And then we reactivate them, and we get, again, more immune cells now in the peritoneum. When we look in the gut, in the colon, nothing. So it seems to have some kind of anatomical specificity. So what we are kind of arguing that is that during inflammation, we have some kind of encoding of sort of immune engrams that are formed of the original experience that can then reactivate it. And anatomical specificity is part of it. There are many questions that emerge. For example, how specific is this effect, right? And the question, and this is something that we don't know, and hopefully Amelia can answer that. And is it only in the insula? I can say that not only unlikely, I know that it's not only there. You saw how many brain areas are activated. The question is, I think that different brain areas, different representations, and maybe I will show a little bit of that. And then there are also questions of what kind of information is actually stored there. Like, what are we keeping there, and how is the signal mediated? So I'm going to talk today about these 2 questions. What kind of information is stored, at least the first clues that we have, and how is the signal mediated? So Megan in the lab, she wanted, in collaboration with Isaac Chiu's lab, we wanted to ask what kind of information can be stored. So how did she approach it? She started with functional labeling. The way she did it, she used the trap system to capture the neurons that were active, but now she expressed there viruses that are going to label either one way out or in all the outputs and all the inputs, and compare how they change during inflammation. And when she did that, she found that a lot of what was changed is the communication with areas related sensory and pain-related areas. So okay, so is pain part of this immune representation? How does pain affect the encoding of the immune response? So what we did is the following. Again, I'm going to show you just small parts of that. But basically, we induced the inflammation. In this case, it was, again, peritonitis. But then we attenuated pain in several analgesias, different analgesics. And what I'm going to show you here is a result from RTX ablated mice, meaning that we kill all the sensory neurons that express 331 and are known to be involved in pain. So we encode in the brain during this time, and then we're going to reactivate. And we're going to see how it affects the immune response. So first, just to show you that under baseline conditions, because the fact that we are killing RTX neurons does not change much the immune response. It's not like they encode something different, a more attenuated immune response. It's very limited. But the reactivation is very different in this case, meaning it seems that at least pain is and everything seems to be mainly attenuated. So kind of what we can start to conclude is that pain is probably part of the information that is encoded in the brain. And what we are seeing is also that all the connectivity, once we have RTX-treated mice or analgesia, all the connectivity of these trapped neurons actually changes with the different sensory areas, and it seems to be part of what is encoded there. Of course, I think what it speaks to also is why pain. And I think of pain as a danger signal, because otherwise it doesn't make sense a lot that the brain will record every immune response, right? There is something has to pass a threshold. And I think part of this threshold should be related to pain. And then the question, how does pain do it? That's a big question. But I think it serves as some kind of indication for the brain to record or not. The next question we can ask here is, how is the signal mediated? Basically, if I'm, again, activated the insula, how come I'm getting a response in the peritoneum or in the colon? So what we did is the following. We trapped neurons in the insula and injected to the colon PRV. PRV is a retrograde virus that will go all the way up to the brain. And basically, through several synapses, will capture neurons that somehow can anatomically communicate with the colon. And this is what you see here. So this is the insula. And the yellow ones are neurons that were both active during the inflammation and via several synapses can connect to the colon. Next, we ask, OK, so how is the pathway mediated? So again, we injected PRV. But now in the brain, we injected AV1, which will jump one synapse forward. And we're going to ask where they meet. And now they meet in really cool places. Actually, the DMV and the RVLM, one controls the sympathetic, the other controls the parasympathetic. Now, it suggests that at least anatomically, the pathway is mediated by this, by the sympathetic or parasympathetic nervous system. We don't know exactly how it does, but we have some more evidence, a little bit, about changes in the tissue after inflammation. So one of the things that we tend to see is that any tissue, after it went through inflammation, doesn't go back to normal. And one of the things that change is receptors to neuronal mediators. So then the idea is, the way we are kind of thinking this in the current model, is that once the brain sends signals to a tissue that already experienced inflammation, this is how it can initiate the local immune response. So it's kind of, in contrast to the classical engrams, it's more a distributed trace between the brain and the tissue itself. So maybe finally on that part, what is the clinical potential here? If we have a way, if we understand that the insula is somehow involved, maybe in triggering even immune responses, maybe if we block it, maybe we can somehow attenuate the immune response. And that's exactly what we did. So we created inflammation. And then, again, we had two groups of mice. In ones, we sham controls, and mice in which we use the inhibitory form of DREADS. And if we are creating an inflammation, so there is an active inflammation, but we inhibit the insula, we get some reduction in the inflammatory response. So what you're looking is the colon. The colon shortens during colitis. So the shortening is not as big, but this is not really amazing kind of data. But I think where it's stronger is actually in the amount of immune cells that seem to be reduced once we inhibit the insula. It's also interesting because it suggests that the insula is active under basic endogenous responses. And what we are doing now is work with Ben Shofty from Utah University, and actually he's a neurosurgeon who has patients implanted with electrodes for epilepsy. And apparently, in recent years, they started to put some electrodes in the insula as part of the procedure. So he's stimulating this area, and now we are getting samples. We already have 13 patients. And I think if I'm kind of thinking in long-term terms how it can integrate is that if we can actually detect aberrant activity in the insula, which is typical of an inflammatory relapse, then we can actually turn down the activity in the area locally by these implants, which is, I think, something that can be kind of a new way of treating these severe autoimmune conditions. So it seems that the brain stores immune memories, which we call them immune engrams. And as I mentioned, we also talk about this distributed trace. It also suggests that the classical idea of immune memory as something that is encoded by memory T cells, antibodies, is limited. And we should think that maybe part of the immune memory is also in the brain. But the problem, of course, that it now, what it does, it creates these relapses of autoimmune conditions. So it says something doesn't make sense. How you explain why we have this feature to begin with. And I think we should think about it like every kind of memory, fear memory. If you have fear memory that keeps you away from the wrong alley, it's wonderful. But now if it's overwhelming and it's now being triggered by many different things, now it can become a case of PTSD. And the same here. If every time that I go to eat in a certain place or drink in this place, I'm going to get salmonella, so of course I better mount the appropriate immune response. I'm anticipating that. But now if different random signals are going to activate this immune response, now I can talk about kind of physiological PTSD. And I think if we play with that, it might be somewhat more relevant. Do I have time a little bit? So in the remaining time, I want to talk about how relevant it is really to humans. And the work that I'm going to present was done together with Professor Talma Hendler from Tel Aviv University, Nitzan, who is now here. So all the questions on that will be directed to Nitzan, and together with Tamar Cohen. So what's the rationale? We wanted to see whether activation of the brain reward system can somehow affect also the immune response in humans. How do we do it? Of course, with fMRI. So now we can actually detect how much activation we have in this area. That we can do. But now the problem is that humans are very different in terms of their immune response. So how can we time lock the immune activity so we can actually follow a change in the immune response that will be correlated to a change in the brain activity that we are looking for? Vaccines. People are actually getting vaccines. So these all subjects were students that were supposed to get HBV vaccine. So now we had the time point of the vaccination. And then we could assess, we have for each subject, we could see the change in the antibody titer. So, of course, neurofeedback, I think, in this crowd needs little introduction, but the idea is that people were allowed to they offer different strategies, but basically they use whichever strategy they chose, and they just got a feedback on how well the activity was. We had 85 subjects, 34 of them got feedback on activation of the mesolimbic system. In this case, it was WTI and the nucleus accumbens in general, so the whole mesolimbic system. Another group of 34 was random neurofeedback, and another group was left untreated. So now the subject come in, so we take in their blood, they are getting into this fMRI, they're doing the trainings, 3 sessions, about 50 trials overall, and then in the last day they come again, do the session, we're taking the blood so we can know the antibody titers, they are getting the vaccine, and 14 days later we are looking at that. So of course, because neurofeedback is rewarding on itself, and in all cases we have some level of activation of the reward system, so what we ended up doing is actually comparing VTA activity or nucleus accumbens activity compared to the antibody titer. And what we see now is the following. So you can see that there is actually a nice correlation with VTA activity, but not with the nucleus accumbens. And why I find it extremely interesting is because it seems, so it was surprising to us, I have to admit. We were sure that we will see correlation with the nucleus accumbens as well. And then Nitzan and Talma called us and said, it's weird, we don't see the correlation. And luckily, the same week, Itai had his data showing, when he did all the different projections, he said, well, actually, the one to the nucleus accumbens is not the one that is doing the immune effects, and the boosting comes from one of the different projections. So maybe retrospectively, we would have done it more focused on the BTA, and maybe we could have gotten even stronger results. But it was actually amazing to see how the two kind of, the animal data and the human data came together at the same week. So, okay, what are the clinical implications of this whole field? And I will sum up here. It's coming together very, very actively. I think the recent, I don't know if you know, recently the FDA approved the first use of vagal stimulation as a way to treat autoimmune conditions for some indications. So it's actually happening now. All these intracranial electrodes, which is again a field that becomes more and more popular, it's another option. Of course, all the TMS and all the other more focused methods. And I think even this crazy machine that we saw back then can become a reality, but just in a bit different way in the modern world. And I think the whole field of virtual reality and virtual games can be a game changer for immune modulations. So with that, I would like to thank the people who were involved, our funding, and our collaborators. And thank you. I'll be happy to answer questions.